Everybody, welcome to the Center of Ethics and Human Values monthly conversations around research ethics. The OSU Center for Ethics and Human Values CARE programming is presented with support from the College of Medicine Center for Bioethics, the College of Public Health, and the Center for Clinical and Translational Sciences. My name is Dana Howard, and I'm going to be moderating. And I'm in the Center for Bioethics in the Philosophy Department at Ohio State University. And I'm so excited to have this conversation about the ethics of human challenge trials. We have two really excellent distinguished guests that bring different levels or different avenues of expertise. We have Professor Seema Shah, who is Associate Professor of Pediatrics at Northwestern University Medical School and is the Director of Research Ethics at Lurie Children's Hospital. Professor Shah is an expert in the fields of pediatric and global health research ethics and has chaired an NIH committee on the ethical considerations in conducting Zika virus human challenge trials. She's also consulted with the WHO on ethical guidance for human challenge trials more broadly. And we also have OSU's very own Susan Koletar, who is Professor of Infectious Diseases and Internal Medicine and is the Director of the Division of Infectious Diseases. She's also the Principal Investigator of the AIDS Clinical Trials Unit at OSU. I'm so excited to have the two of you and to jump right into this. Let me just tell the audience what to expect about the next hour and 15 minutes. We're going to start with Seema, who's going to present from sort of an ethical guidelines perspective, some of the main ethical challenges, what is distinctive about human challenge trials, but also maybe not distinctive, but what is sort of like made very salient with human challenge trials. And then we're going to have Sue discuss like from her own 30 years of experience in the field, conducting these trials, what challenges have you experienced and how you've been thinking about this ethically. I then have a set of questions that we're going to ask, but it's also, we're going to have plenty of time, the last 25 minutes or so, from questions from the audience. So if you see at the bottom of your screen, there's a Q&A function. As we go throughout the whole conversation, please feel free to input any questions that you may have, and we'll get to those shortly. All right, so to no further ado, Seema, you can take it away.
Hi, everyone. Thanks so much, Dana. It's just such a pleasure to be here and have this conversation with all of you. So I don't think I could share my screen at the moment. I don't know if, Dana, if you can make me a co-host or enable screen sharing, but let me not waste any time and just start talking about let's see if it works now. Oh, yes. Excellent. Okay. Can you all see that? Yeah.
And we can see it. Thank you.
Sure. Sorry, I've got a lot of windows that are popping up. Okay. So I'm really delighted to talk to you about ethical considerations for what I refer to as controlled human infection studies. I think historically they were often referred to as challenge studies. And what I'm going to do is first give you some brief background on what these studies are, just to make sure everybody's on the same page. Then I'll talk about whether they're ethically unique, what the salient ethical considerations are for these studies, and finally, I would like to focus on some lessons we can learn from how these studies were used during the COVID-19 pandemic. So first, what are controlled human infection studies or models? These are also referred to as human challenge trials, and they're studies where people are deliberately exposed to a pathogen, and this is done for scientific purposes. So it can be to understand more about the early phases of diseases when people don't usually present to care, or to test vaccines or treatments, or learn more about diagnostics. And you can see these two pictures illustrate participants in these studies. So the first one is a man who participated in a cholera study in the University of Maryland. And the reason he's holding a whiteboard with the number 26 is because that's how many liters of intravenous fluid he needed. So I think this illustrates that these studies can be quite burdensome, but in this study, there was no concern about any long-lasting effects, and as long as participants got the treatment they needed. The second picture is from a controlled human influenza infection study that was done at the National Institutes of Health. And for these studies, there is no specific treatment that's offered that can definitely cure influenza. So these are actually some of the riskiest controlled human infection studies that are done. And what's really important to remember is that there's a difference between the first step, which is creating a model where you have to find the right dose or the Goldilocks dose that can infect the majority of people in the trial, but not make them too sick, and develop all the parameters of how you do this safely, reliably, and robustly. And then once that's been done, using that in studies that can test vaccines or other interventions. So when we think about these studies, they're relatively small and therefore can be relatively fast to conduct and very efficient once you've created a model. But creating a model can take somewhere between 4 to 12 months. They're capable of rigorously addressing a wide range of research questions, which include investigating the mechanisms of early disease, as I mentioned, understanding transmission and immunity, and gathering preliminary efficacy data on investigational vaccines or treatments. And one critical thing to point out here is that they're usually not the last stop in testing. You typically need to gather more data in a wider group of people, since these are relatively small studies, before you know that a vaccine will work in a larger population. And I think it's important to note what some of the prominent successes have been, because sometimes when I give these talks, people sort of ask, do these studies really matter? How important are they anyway? Well, for the malaria vaccine, these studies served as the proof of concept for the first malaria vaccine that was approved and is now in use around the world. And, what's also interesting is that in malaria in general, these studies are sort of a part of the pathway where anything that's going to be taken out to field trials has to pretty much go through controlled human malaria infection models or studies first. They also supported the FDA licensure of a cholera vaccine. And this was when it was impossible to mount field trials where people tried to do it out in the field and couldn't actually catch outbreaks when they were happening in a way that would be able to show whether the vaccine worked. They established the correlates of protection for influenza and accelerated approval for a paratyphoid vaccine that was recently approved. And the key point to remember here, though, is that these studies really have a very long history, and they were part of the start of what we even think of as vaccination, when Edward Jenner used a cowpox and then smallpox challenge of his gardener's son, James Phipps, as far back as 1796. There have been a lot of really seminal challenge trials that have been done, and then challenge trials that really raised ethical issues, like the Willowbrook pediatric challenge studies, which were hotly debated in the literature. But it wasn't until fairly recently, in 2001, that the first article on the ethics of controlled human infection studies was published, where there was really a sustained analysis of the ethical issues. And this was done by Frank Miller and Christine Grady. So, the key to take away from this is that despite this long scientific history, ethicists haven't really focused on these until relatively recently. So one of the key questions that comes up for a lot of people is, are these studies really unique and different from other types of research? And a lot of people have a concern that because these studies involve the intentional infliction of harm on participants by doctors, that's something that's really ethically unique. When you look at views among ethicists, Miller and Grady in 2001 argued that these are no different than phase 1 trials with healthy volunteers, because in phase 1 trials with healthy volunteers, people are not sick and they're given medications to see if those medications are safe, so that those medications can later be used for healthy people. So the same balance of exposing some people to risk for the benefit of future patients in society, that same issue comes up in controlled human infection studies and phase 1 trials. But by contrast, Banbury and colleagues argued that controlled human infection studies are morally problematic because of this intentional infliction of harm by researchers, because of concerns about public trust if these studies were to be done, and because of the worry that other people outside of the study who didn't consent to being exposed to risk might also be exposed to risks from infection. So I think it's worth looking a little more closely at this question. And when you do, you realize that there are actually several different things that you might think are ethically unique about these studies. One is, as we've mentioned, that researchers intentionally directly cause harm to participants. Another is that there's uncertain benefit to society from these studies, that they really are, a unique part of the research pathway. A third issue is that they expose participants to high and uncertain net risk. A fourth issue is that they expose non-participants or third parties or bystanders, whatever you want to call them, to risk. You might worry they violate the right to withdraw. And then some people are concerned that they pay a lot of money and that could compromise participants' judgment. But as you look more closely at all of these things, there are other examples in research that do the same things. So for instance, if you think about the direct cause of harm to participants by researchers, phase 1 studies with healthy volunteers involve exposing people to harm, as I talked about, and also any study that's done with procedures that are only done for research purposes. So if you do a blood draw or, even up to a liver biopsy, which is one of the riskiest procedures that IRBs sometimes allow in research that's not for someone's benefit, all of these things involve the direct cause of harm to participants. With regard to research having an uncertain benefit to society, Dana had a really nice example of stuff she's working on related to studies that are trying to address policy that's informed by politics or ideology. So specifically studies about abortion reversal. And anytime you're doing a study that's designed to reach policymakers, there are always questions about whether that evidence will actually move the needle and what benefit may occur for society. When you think about exposing participants to high and uncertain net risk, HIV remission studies, which were initially referred to as HIV cure studies, it really took things like chemotherapeutic agents and gave them to people who were well controlled on their HIV medication. And the goal was to achieve this remission or cure of HIV, but these studies did involve very high risk, at least initially. When you think about non-participants or third parties, there are tons of examples here, but one that I think is really interesting and increasingly being used is when data is collected digitally, where participants have wearables or in their homes, because these devices can pick up information about other people that may have to be reported or may reveal things that these people would prefer to keep private. When you think about the right to withdraw, you have biobank research where samples are stored and everything's sort of mixed together. Sometimes it's very difficult to withdraw your sample from that study. And there are other examples where we don't just let people withdraw from research right away, which is something that could come up in controlled human infection studies if somebody is enrolled in a trial and is infectious and wants to leave. And finally, when we think about payment, what's interesting is a controlled human infection study researcher I've collaborated with says that she struggles to compete with phase 1 trials, and that many of the people she tries to enroll would prefer to enroll in phase 1 trials because they get paid more. So if you look at the controlled human infection studies done with SARS-CoV-2 during the COVID pandemic, they paid the equivalent of $6,200 in the US, but phase 1 trials can pay as much as $13,000. So what I think is going on here is that these types of studies are not unique, but they raise a constellation of issues that we haven't fully resolved in research ethics, and they raise them all at the same time. So when that happens, I think there's a lot more complexity ethically that researchers, IRBs, and others have to grapple with. And these include the things we've just been talking about. The only thing that I would add that I didn't mention is this idea that Banbury and colleagues came up with, which is that there's this potential for public mistrust. People have that reaction of, It seems wrong to expose people to a disease when they're not sick. So what I think is also a really critical issue that's often disregarded is that when you're creating a new controlled human infection model, that's different from using an established one in a controlled human infection study. And because there are higher levels of uncertainty and ethical complexity, that means that you need stronger justification to navigate the cutting edge of it. So I want to talk now about the ethical guidance. There are a few different documents. The World Health Organization, I consulted with them for the development of these guidance documents. There are key criteria for the ethical acceptability of COVID-19 human challenge studies. And then there's the WHO guidance that's more general on the use of these studies. I've been leading a multidisciplinary international group to address these issues prior to the pandemic. And so during the pandemic, we took our framework and applied it to COVID-19. We also wrote a special issue diving in more deeply into some specific issues and concerns. And I think these are, if you look at these documents, they have different numbers of criteria and they have different labels for the criteria. The WHO guidance document really focuses on also procedural protections, doesn't address payment, but I think these are really similar, and I'm going to sort of try to distill the criteria that I think are common across these two frameworks so that we can have a kind of core set of criteria to work with. And when I did that, I found that, these are the 7 criteria that you would think about substantively, and then there are separate criteria for procedural issues that we can talk about at the end. So first, social value. This is the idea that the research has potential benefits for populations, and we have to think about the magnitude, distribution, and likelihood of those benefits. And for creating a new controlled human infection model, it's really important to be able to delineate a path from that to health benefits, but it can often be a long path and one that has a lot of uncertainty in it. When you think about a reasonable risk-benefit profile, it's important, as with all research, to ensure that risks and benefits to participants should be systematically identified, evaluated, and minimized, and below some upper limit of risk. But you can do various things that, for controlled human infection studies with SARS-CoV-2 that involved enrolling young, healthy people at low risk, monitoring them closely, and then figuring out what treatment to give them. But that isn't necessary if enough is known about minimizing risks, as was the case for cholera studies, for instance. Third parties also raise challenging issues, but it depends on how the infection spreads. And what typically happens is people are confined during the infectious period, and research personnel need personal protective equipment. The longest study I've ever heard in terms of confining participants was 29 days. So it can be quite challenging and burdensome to be confined for that long. A third issue, which we don't often talk about in research ethics, is appropriate site selection, which I think has a number of different factors that matter, including, the local expertise in treating the disease, the feasibility of recruitment, and what people think about controlled human infection studies. A fourth criterion is fair participant selection, and the key issues here are to ensure that clinically relevant populations are included, and most importantly, that people who would face unacceptably high risks are excluded, including if the risks to third parties might be an issue. So sometimes for certain types of studies, like studies with RSV, people who work with young children or have young children in the home would often be excluded from those studies. Community engagement, I think, is a critical issue for controlled human infection studies, and it can vary depending on the context. I think when you're developing a new controlled human infection model, it's important to have heightened levels of engagement. And it can be useful to consider embedded social science research as well to understand what participants are thinking or what issues are coming up in trials like these. This is a really nice example from Dorcas Kamuya and colleagues of sort of developing a deliberative model of community engagement where there was a lot of back and forth between the community members and the researchers, including about rumors that started to spread about these studies during. And as always with research, informed consent should be robust, but evidence-based methods are critical here. I think also there are key elements for these studies, including exposure to infection, that there are burdens and risks for participants and risks to third parties, and any restrictions on liberty. In this attached article, we also argued that you shouldn't necessarily have blanket restrictions on people with low literacy or education levels, but I'm happy to talk more about that if people are interested, as it is something that different people debate. And finally, payment has been really controversial in controlled human infection models. And these are some of the examples of the types of controversy that have arisen, with some people using robust payment models, others arguing that you really should just pay a lot and pay for as much risk as people are taking. I think participants should be compensated for time and expenses to avoid exploitation, but they should receive treatment or compensation for injury if it occurs, and not just be paid for risk up front to really focus on those who were harmed. And finally, I think one other issue with payment is sometimes people worry about undue inducement, or if people are paid, they might withhold information that makes them ineligible. And I think there are other ways to address that, including by having a robust informed consent process. So with regard to the idea of procedural protections or specialized regulations or review, I think a lot of people agree there's no need for special regulations, but there can be extra guidance that helps refine how IRBs or research ethics committees apply those regulations. And the WHO recommended specialized expert review for the controlled human infection model that was developed during COVID. And I think what that reflects is that when you're developing a new model, it may be useful to have a more robust and rigorous process to address all of the special issues that come up. So I'm just going to briefly address some lessons from what happened during COVID-19. You all, if you were following this, may be aware that the UK was the first and only country that did these studies, even though several countries thought about it. The US developed the strain for these challenge trials, but Tony Fauci called it a plan C or a plan D if the standard approach to vaccine trials, which you'll hear about in a minute, I think, from Susan, didn't work. And what the UK did was they did two different controlled human infection studies, one with people who'd been previously infected and another with people who were naive, and then have done subsequent studies with new strains. And there is now increased interest in this for future pandemic preparedness. So one interesting thing is that in the UK, they had a specialized committee that did training exercises before they even reviewed the protocol for SARS-CoV-2 chimps. And this article provides some detail about that. And the lessons that I think were learned were that engagement and context matter. So in the UK, they had groundwork that had been laid to develop the infrastructure and the understanding about these types of research. And there was also universal healthcare, which we don't have in this country. And that meant that any long-term health issues like long COVID could still be addressed, even if they emerged after the research was over. However, uncertainty in these trials remained high and I think problematic. The initial dose that was given to participants was 10 times lower than what the WHO had recommended, but was still the right dose to infect most participants. And the long-term effects of COVID at that time were not that clear and have only recently become more clear, although there's still a lot we don't know. Finally, the findings had limited generalizability, partially because they needed to exclude people at high risk of severe disease, but I think most importantly because we're always chasing variants when it comes to COVID, given the amount of time it takes to set up the model. So what I want you to take away is that there's a complex history here, which really is important to take account of, and these studies raise a constellation of unresolved ethical issues, especially when you're creating a new model, and that may benefit from specialized guidance and review. And finally, I really encourage all of you to sort of think more broadly about how, as we look at these types of studies, how we might develop new insights for other types of studies where they are on the cutting edge of ethics. So I'll just thank my colleagues who've helped me with this work and funders who've supported it. and turn it over to Susan.
Thanks, Seema. That was a great overview. I'm going to take a little bit of a different approach, one without slides, and two with the disclosure that I am not an ethicist or a lawyer. So, but I'm a clinician at heart, and so I just wanted to maybe take a few minutes, and because I really want to leave time for questions, to talk about clinical trials in general, and and I learned early on, or appreciated early on, the dilemma of any clinical trial, human challenge trials included, is that your hypothesis has to be something that you believe in, but not know, right? So as a clinician doing an intervention trial of any sort, you have to have enough preliminary evidence, whether that be in a test tube or in animal models, or sometimes things that you have seen in the context of clinical practice, to think whatever you're doing, whether it be a vaccine or a treatment or a procedure, is better. And better is loosely defined, right? Better is more effective, or maybe more tolerable, less side effects. You had to, and there's lots of examples of that we could talk about. But you have to believe that that's true, and you have to not know, right? Because once you know, then you don't need to do the trial. I had in the early part of my life, I was also the chair of our IRB. So I guess that's my other disclosure. I believe I have read the Belmont report and the Code of Federal Regulations at least 100 times. So, but that's the basis of clinical trials, and the other thing that's a little bit different about the challenge trials is that you, well, I don't know if it's really different, but the gold standard in terms of moving interventions or treatments forward in medicine has always been a randomized, double-blind, placebo-controlled trial, which again allows for gaining lots of knowledge. So I think the closest, and Seema already said this, I think the closest similarities between challenge trials and other clinical trials are the phase 1 studies, where you take by design healthy people, relatively few numbers, and that's a plus side, right? Because you can move things more quickly, and you can hopefully gather enough information, but they're really designed for safety. I've had discussions in years past with some of my oncology friends and some of their phase 1 clinical trials, which again, by design, often will be done not necessarily in healthy volunteers, but in people who are experiencing sort of the ravages of malignancy that don't have any other options. And that's, again, when you think about clinical trial designs, the driving principle is minimizing risk and maximizing benefits. And so if you have a toxic chemotherapy, it's really hard to justify giving that to a young healthy volunteer. If you're trying out a new vaccine, a new antibiotic, or a new blood pressure medicine, or a new vaccine, healthy volunteers really gives you some cushion, I will say, to put that risk-benefit ratio in the favor of benefit, as opposed, or at least do no harm. And I think that is an important thing. I'll digress for a minute and tell a quick story about one of my early phase 1 studies that I did with an antibiotic in conjunction with some HIV medicines, and it was a treatment for tuberculosis. And some of the early days of treating HIV with the protease inhibitors, there's lots of drug interactions. And so we thought, well, what if we do this? What if we can overcome that drug interaction by giving double the dose of this particular agent? And so we gathered up some healthy volunteers who met all the criteria, and we put all on one day 3 people in this study, many of whom I knew personally, and they did great the first time around, and then as we kept ramping up the dose, they all got jaundice, they all started throwing up. I mean, it was a horrible day in my life, actually. but I tell that story because one of the, and again, Seema talked about this a little bit, how do you reconcile the management of untoward effects in any type of trial? So the poor guy on the cholera pot, he got 26 liters of fluid. But if you have something, if you are in a trial where you're being challenged with malaria, well, there's opportunities for intervention and control the disease. But that's always a major major thing. I did want to touch on a few other things and maybe then let it open for question. I think the idea of challenge trials and the impact they have also on the participants surrounding family, community, and stuff is very is an important one, and we are struggling. That sounds dramatic. We are dealing with that now in terms of the HIV remission/cure studies, where we're looking at treatment interruptions. And so one of the great coups of modern medicine has been gaining the knowledge that controlling HIV, having undetectable viral loads, essentially results in no transmission. So if you're doing treatment trial, if you're doing cure studies, and you purposely tell someone, again, so this goes back to do no harm, or how do you do this? You tell someone to stop their medicines, hoping that they won't become viremic again, but knowing that that's a possibility. How do you deal with them and their potential partners and the community at large? So I think there are some similarities. And then I did want to make one other comment about some of the payments, because I think we struggle with that in clinical trials all the time. What is coercive versus what is fair compensation? And I think there's really a lot of local context to that as well. And it's really hard when you're doing particularly international studies to really appreciate the local context. And I think that's something that probably is worth a little bit more discussion. And even in some of the trials we do now, there's I believe they're called conditional economic incentives, paying people for actually reaching or taking their medicines and doing the points. And so that's another aspect of this. And then finally, and I'll quit rambling on here, the COVID-19 vaccine trials was such an interesting time in all of our lives. I think we should just take a moment and say, wow, how did we manage to get through that? And I was, before we started today, I was pontificating about the impact that the early HIV studies had on the COVID pandemic, because our knowledge of virology and immunology expounded and expanded exponentially over the last 30 years, that we were able to go from identifying the virus quickly in terms of sequencing the entire genome, to having tests available, to having treatments available. And that's the other thing, that and I don't know, there's relatively not tons of press about the early challenge trials, rightfully so, right? Because everyone was afraid and we didn't know really what we're doing. And although we know more about the long-term effects of COVID, we don't know all of them. I'm convinced of that. And so how those played out in terms of the vaccine trials that were done, well, first of all, it was an amazing feat among clinical trialists and participants in clinical trials across the board. And before I forget to say this, I think we all collectively owe a huge debt of gratitude to anybody who participates in a clinical trial. We could not have made the advances in science without people who have been willing to volunteer either for a challenge or for one of these trials. And the trials, the COVID vaccine trials, were successful so quickly because one, there was so limited immunity across the world to COVID. So it was not actually a challenge trial per se, but it was just a challenge of living per se that got so many people exposed. The powers that be threw enormous amounts of money into setting up these trials. We did one of the vaccine trials here, and I could tell you it was a whirlwind. We tried to get all the IRB issues taken care of quickly, institutional biosafety. We put a couple hundred people in studies within like a 2-week period of time. So it was an enormous effort. And I think because of the lack in general of immunity and because of the efficacy of these vaccines, we were able to come to decisions pretty quickly, which leads me to my perhaps final provocative point about challenge trials is that in regular interventional trials, when you have something that is known to be effective, it I dare to say borders on unethical to do placebo-controlled trials if you have the opportunity to use a standard of care. Challenge trials where you're trying to figure things out, makes it a little bit murky. And maybe I'll stop there. And I know, Dana, you have a bunch of questions, and I can't tell if there are any.
There are some questions already in the chat, which is great. So please, participants, please continue asking questions in the Q&A function. I'm going to start with some questions, and then we're going to already jump into questions from the audience. I really appreciate Seema just giving us sort of ethical guidance framework for thinking about all the different ethical considerations and, you know, really thinking about what is distinctive, what is not distinctive, but like, you know, really a constellation of the issues. And I really appreciate Susan bringing up your own experiences and the ways in which we tend to think about like the young, healthy volunteers as like the lowest risk people, but thinking really sort of being really thoughtful about what we mean by risk and when different populations might be like better candidates for some of these higher risk studies, I think is a really important thing to consider and does complicate the ways in which we're thinking about these issues. So let me start with one question about COVID-19. Susan, you brought up the AIDS crisis and sort of COVID-19 crisis and the fact that we sort of, you know, all the sort of the heightened stressors of being in those emergency situations. And one of the things that we see in these sorts of emergency situations like COVID-19 and Zika also was an example of this, is this sort of growing public advocacy for human challenge trials in these situations. And I'm wondering if you think that there's something exceptional about these emergency circumstances that make research like human challenge trials more ethically appropriate when they wouldn't otherwise be ethically appropriate in other situations. So I have no idea what this is. It's like an open question. So I would love to hear your thoughts. And is there something special about emergency situations that call for or allow at least for higher risk research than, you know, in standard times may not be allowable? So maybe we'll start with Seema and then Susan, you could add on.
Yeah, that's a great question. And I'm going to fold in one of the questions from the audience into my response. So I think what often happens in emergencies is we're caught unprepared, right? We don't have a model on the shelf that we can pull off and say, you know, for this new pathogen that is now circulating, we have a controlled human infection model that we can use. So we have to develop the model. And somebody asked, you know, what's the difference between a controlled human infection model and a controlled human infection study? The idea is the process of developing a model can take 4 months to a year. And the idea is to find this Goldilocks dose, right, that infects most of your 15 to 150 people who are in your study, but doesn't make them too sick. And then, you know, you have a strain that you know is just the pathogen you're you're concerned with, not also other things that people might have. And you can give that dose to people, and you know exactly, how to set up the facility where they're getting their the doses and without infecting research personnel, and then also how to treat them or manage them once they get infected. So there's some things where we've been doing that for a long time, like malaria, and it's a very safe model with a lot of data on how to use it. And there are other things like COVID where we don't know a lot about it. So in general, I think most ethicists believe that in an emergency, you can do things that are more risky if there is corresponding value. So if there is a good reason to think that doing something that is pretty risky could benefit a lot of people, that is something that most people think we should do. I think there are a couple of problems with that, somewhat practically and somewhat, conceptually in an emergency. The one big practical problem I see is sometimes we're not very good at judging what is of high value in an emergency for lots of reasons. One is like, we're all, in panic mode. So it makes it harder to make careful judgments. Another reason is that there's a lot that goes on in an emergency. So many people are marshaling every resource they can to do things. And, the way that vaccine trials were done by people like Susan, that was unprecedented to get an answer to the question of, how can we prevent COVID-19 or at least prevent severe disease? To do that in just about a year was remarkable. So I think one of the challenges is that I'm not sure that controlled human infection models are the best thing in that rapid emergency response. And I don't think that the COVID pandemic showed that they could be as useful. I think the area where I have more optimism is about maybe we can get better at preparing for future emergencies. We can start to understand, families of viruses that keep coming up in these pandemics. And we've had 3 coronavirus epidemics that have gone around the world now, and we should really understand this family of viruses better. So maybe we can use controlled human infection models to do that. But I think in an emergency, we still really haven't seen a really good example of how these models can be useful. And most of the examples that I cited that where they have been really useful have really been outside of the emergency context.
Yeah, and I would add that one of the driving factors in the clinical trials world, for those of us who do it all the time, is the concept of informed consent. And I think that adds another layer of complexity. Informed consent in emergency situations, even if they're not public health emergencies, is a whole nother beast. And so, now informed consent's only as good as the information that we have to tell people. And so that's the other little bit of a tricky thing here. And then finally, I will say, I have been impressed what motivates people to participate in trials. And that's probably has its own ethical constructs as well. But now again, I'm just telling stories again, but I asked everyone because I saw almost everyone who participated in our vaccine trials, and some of it was purely altruistic. Some of it, which struck a chord with me, was participants from typically underrepresented communities who wanted to make sure that their community was represented. And some of it was because people were afraid, and they thought, even though we were good about informed consent, and this is a placebo-controlled trial, they thought getting a vaccine earlier would be better for them. even though I said maybe not, but okay. So
thank you. Yeah, so one thing that you mentioned, Seema, in your presentation, and I'm interested just to focus in on it a little bit, is this sort of do no harm idea. So, when it comes to clinical trials, in particular vaccine trials, we expose participants to risks all the time, right? And people tend to think that as long as the benefits of the research outweigh the risks, then it can be ethically appropriate. And I'm wondering if you think that there is something that is ethically distinctive about the types of risks that are involved in human challenge trials. So, the particular exposure, intentionally exposing people to a virus, is that a different sort of harm than other kinds of risks that are incurred in phase 1, say, trials? Maybe we'll start with Susan, and then Seema, you could go afterwards.
It's a complicated, it's a complicated question. Again, I'll go back to, I think the informed consent part of it is the most critical, and what you think the potential risks are. I think one of the inherent risks in human challenge trials is that you don't really know what all the risks are. I mean, obviously the risk, there's layers of risk. There's probably the risk of getting the disease that you're trying to prevent, right? And that's true whether it's a vaccine trial or other prophylactic trial. I mean, there was a lot of studies done with malaria as well, using chemoprophylaxis and stuff. And so where then the risk is both the risk of the disease itself plus the risk of the intervention, whatever that might be. Sorry.
Yeah, I think it's a really interesting comparison. If you look at a field trial where you're trying to see if a vaccine works, right? So there is this intuition that with a controlled, so let's say you've developed your controlled human infection model, and you're taking that model and you're trying to understand whether your vaccine works to protect people from getting infected. So then you're applying this model in a study. And what that involves, right, is taking half the participants, giving them a placebo vaccine, half the participants giving them the actual vaccine, and then using your method of deliberately infecting them and seeing what happens. And in some ways, that's not that different than what happens in a field trial. And I think critically, in terms of the intention of the researcher, it's not necessarily that different, because no researcher wants participants to get very sick, right? That is not good for anybody. But what both researchers who are in these controlled human infection studies and researchers who are doing larger field trials, what they want is for there to be infections that occur so you can see a difference between the placebo arm and the vaccine arm. And in COVID, I think one of the things that really helped trials along was that there were so many infections in the United States, right? The amount of people getting infected helped us get to an answer faster. And so I think in that way, I don't actually think there's that much of a difference between the intent of the researcher or the fact of what's happening in the trials. I think the thing that keep in mind is that it feels very different to people. So some members of the public may feel like this is really not what researchers should be doing. And that may be based on sort of a failure to really understand what's going on in research. Mm-hmm. So I think that suggests that we need to do more education around these models and research in general, and that sometimes having that public engagement prior to developing a new model can be really useful. And then I think the other thing that's important to remember is there are lots of different kinds of models, and some of them I think kind of raise this ick factor for people a little bit more than others. So one of my collaborators is somebody who does controlled human infection models with schistosomiasis. And this is a parasitic worm. And so people have to drink a fluid that contains male parasitic worms, and only the males. They don't reproduce in the participants' bodies. And then they receive treatment and they end up being fine. But for most people, that just sounds really nuts, right? Why would anyone do that? And how is that ethically acceptable? And yet it's this neglected tropical disease that really should be addressed, and it's a safe way to do it. And the more you learn about the model, the more education you get, the more I think many people think it can be ethically acceptable. So I think we have to take the public reaction to these models and studies seriously, but I don't know that it reflects a deeper underlying ethical concern.
Well, and when you get down to it, the models and the challenges, they're relatively controlled. I mean, you still, I think you made the point, Seema, you can't truly replicate exposure in the field. And then I think that's one, one of the we did we did an early HIV vaccine trial here, which again wasn't a challenge trial because that would have been unethical. But you're right. Not you hope, but you expect that there will be some natural acquisition of disease. And so those trials had lots of reviews and lots of counseling and despite all that, still didn't work. But I mean, I think you're right. The education of the public is critically important in terms of what is the intent, what are the controls around it as best that we have, and then how do you assure how do you assure that the participants appreciate that so they also are engaged in the process, right? And I think that's another critical part of it. And
So
can I just give you a little bit of historical thing? I think Edward Jenner was always credited for that and certainly did lots of work. But I think I read some historical account that he actually himself as a young child was inoculated with smallpox, and grew up to
there was long history of inoculation, which actually started in Asia and Africa and was brought to Europe, relatively late. So it was, I think there's a historical context that's really important to acknowledge for those studies.
But I think it's also important to acknowledge that this may sound a little Pollyannish. I think the intent is always to try to make something better and understand something more, and that's where you have to try to sort of delve into the details about how to make that happen.
So we are 25 minutes to the end, and I want to make sure that we get questions from participants. So keep on asking questions, and I'm going to fold in Frank Miller's question into this final question that I want to pose to you. And it's exactly this sort of point that Susan was just mentioning about trying to get, trying to advance knowledge, designing studies in a way that is most likely to do that. And you can't know in advance, right? That's the nature of research. So it's always a little bit of a gamble. And so Frank Miller's question is just, was actually there any valuable scientific knowledge that developed from the 2 UK coronavirus challenge trials? And I mean, I'm sort of wanting to fold that into this question about public trust more broadly. So, I mean, we've seen that human challenge trials, they have this long history. They've led to major advances in the development of drugs and vaccines, but there's also some well-documented historical examples of human exposure studies that were seen as deeply unethical and violated human rights, such as the Guatemala syphilis experiments of the 1940s. And so how should it, I mean, one thing that you mentioned, Seema, is just sort of like public education about what research is. How should we fold in these sorts of past issues, like past historical ethical crises of research, into thinking about current ethical assessments of human challenge trials? Yeah, I'll just stop there, and maybe Seema, you could start, and then Susan, you could go next.
Okay. Yeah, this is a really great question. Lots of complexity here. So in general, I mean, I think we can't, I know there's some people who don't see the value of history, who think that sort of, what happened in the past was in a different era. We now have IRBs, we now have protections. We don't need to be as worried. And I think it's a very privileged view of, how you think about research. I think for a lot of people, there's kind of a line that they can draw from prior historical events of exploitation of different groups to, current, the continuation of racism that's still a part of our healthcare system, and, different ways that marginalized groups experience delivery of care even today. So I think for some people, it's in the past, and for some people it's still part of the present. And that, I think, is something we have to take seriously and recognize that as we think about these studies, there are different publics who are going to get different messages from them, and we have to be aware of that. When we I was part of a committee that oversaw, whether controlled human infection studies with Zika virus should proceed. And one story I like to share is that there was someone who was enrolled in a Zika virus vaccine trial who said she didn't want to participate in that trial any longer. And this was just a trial in the field where they were taking a vaccine, people randomizing people to vaccine or placebo and waiting to see if they got the virus. And she was concerned that the injection included Zika virus because she read an article about how we were reviewing the ethics of those studies, where the reporter didn't get it quite right and she misunderstood it. And so that, I think, is really, there are these off-target effects that I think we have to pay attention to. At the same time, we shouldn't just worry about a parade of horribles that we don't have evidence about. So I think we need to do a better job of collecting evidence about how do these trials filter down to people, what kinds of things do they make people think about research in general. And then very concretely to Frank's question, which I think is a really important question, I have really struggled with this, and I've talked to a lot of people to understand what's the best case for what these trials were able to accomplish. And what I've heard from different people is that these trials were really useful in evaluating diagnostics. So using rapid antigen tests, the trials found that, if you keep testing, you're likely to catch over 90% of infections. And that is something where, a lot of people probably in this webinar are very familiar with the process of testing yourself multiple times to see if you have COVID. And that was really validated in this trial. You might still ask, could that have been proven some other way? And there were lots of other designs, I think, that might have been able to help address that. A second thing that people talk about is that there was some finding that prior immunity was actually protective against subsequent infections. And so that finding really led to the UK's decision to kind of reopen the country in the summer of 2021. However, that decision has been criticized by many people, and it wasn't necessarily universally considered a success. So those are two of the issues. Another that people say is that these trials were supposed to help refine a correlative protection. But that there's some data I've seen about this, but I don't think that we've really gotten to a point where we do have correlative protection, and Susan can probably speak more to this. So I'm not sure that we've seen that. So I think right now, these trials haven't really lived up to the promise of what people said they could do. Whether in the future that would be different in a different pandemic with a different scenario, that remains to be seen.
Yes, Seema, I think you answered that really nicely. And again, COVID, it was such an interesting time because things happened so rapidly. I think those trials might have been more useful, perhaps, if there was not such rapidity of vaccine response. And between Pfizer, Moderna, AstraZeneca, Janssen, Novavax. I mean, there were literally, most of those trials had 30 to 40,000 participants. So you got to, and across the world. That's what was really pretty fascinating about that. And so, yes, the diagnostics, I think they probably did help a little bit, but I'm always impressed at how resourceful people are. I mean, people like dove in right away and started looking at diagnostic testing, and which was better. I'm still not sure we've all had that. And my hoarseness here, just as an aside, is a cold. I tested myself for COVID 4 times last week, so I'm not immune from acting crazy. But I think so that was important. But I think because everything else was going so quickly in parallel, maybe the benefits were not reaped quite as much as had been hoped. So I think that makes a difference. And let me just go back to the historical travesties, if you will. I think we just have to address them as they come up. I mean, I've enrolled multiple different people in multiple different trials, and some people are still very concerned.
Yeah.
And I typically ask people, well, do you have any concerns? What's not making sense? So I'll go back to we have to use the informed consent process as just that. It is a process that takes time and is iterative and ongoing. And I think some of the concerns early on about, I think you said it, Seema, somebody didn't want to do the Zika vaccine because they were worried that they would get Zika. I mean, that was a big concern people had with COVID. I mean, those were, you know, I used to tell my grandmother she couldn't, when she told me she got the flu from the flu shot, I'm like, Grandma, that's not true. But I think again, I'll go back to the education and informed consent is a critical part of getting these things done.
Can I ask a question of Susan related to this?
Of course.
So one thing that people have said for the COVID vaccine or COVID challenge trials is that for future vaccines, we could use these controlled human infection models to test them and try and prioritize them. And, you know, probably others in the audience may not be as aware, but there are a lot of different vaccines that are still in the pipeline. You know, there's concern about, can you actually just prevent infection altogether as opposed to preventing severe disease? Intranasal vaccines are another thing that could be really beneficial. We know that the vaccines we have that are effective do have side effects and kind of knock people out for a day sometimes. And so it'd be nice to have a vaccine that had fewer side effects. So I'm curious whether we really could use controlled human infection studies for that, or whether, you know, we could just test those vaccines out in the field since COVID is still circulating.
Yeah, no, I think that's actually a really interesting idea, and it would require cooperation from multiple entities to do that, right? But that would be an ideal. It'd be a great opportunity actually, because there are so I'm, as you might guess, I'm a big believer in vaccines. I think they are particularly safe. I think there are there clearly are side effects, and the long-lasting immunity I think is important. I don't think we honestly know how many boosters one needs. I think the only thing that's been that most people feel good about is that one shot is not enough, but everything else is up in the air. So yeah, no, I think that's a great idea, and if that could be even looked at going forward to potentially assess the impact on variants, you know. But I mean, it's a natural, in many ways, COVID wasn't that different. It's a coronavirus. It's primarily spread by respiratory aerosolization. You know, it has sort of a natural history of disease, and it's almost always true that people who are young and healthy do better than people who are not healthy and usually older. I mean, those, and that's how I started most of my conversations with my peers and colleagues here, is that there are things that we know are true. Let's, step back and try to put that into some context. So yes, so can we do a challenge trial, sort of challenging the status quo? I think that's actually a really interesting idea.
So one final question from the audience that I want to make sure we get to is getting into the financialization of and compensation for these trials. So the audience member asked, what financial incentive amount have you seen that optimizes the number of participants? I'm going to also just ask the sort of ethical issue, since Seema, you mentioned it, but I'm also really interested in, Susan, how you've been thinking about paying subject participants. And one thing that you mentioned, Susan, is, some people are participating for altruistic reasons. Other people for COVID vaccine trials were participating because they wanted to have some possible chance at a COVID vaccine earlier, like, should we worry about financial incentives as somehow posing ethical challenges to research recruitment in a different way than these other potential incentives that people may have to want to participate for research? So maybe give Seema an opportunity about financialization, and then Susan, you can sort of, tell us what you think about paying research participants.
So I will say, I think, I don't know how many times people have done studies to figure out what the right amount is. The controlled human infection studies I've seen have ranged, the payments have ranged between $2,300 for controlled human malaria infection studies to the $6,200 ones for SARS-CoV-2. And I think the other thing, we've looked at what the motivations were of all of the people, right? That over 38,000 people signed up to participate in these trials. So we did a survey of about 1,200 of them to see what were their motivations. And, most of them really expressed altruistic motivations. And then when you look across other kinds of trials, even for phase 1 trials with healthy volunteers, people tend to have some altruistic motivation, even when they might have a monetary motivation. So I think people have complex reasons for doing what they do. The other thing I should mention, we did a study of people who were in controlled human malaria infection studies where it was qualitative studies. So we got to really ask them some questions. And one participant said he really liked new experiences and he had never had malaria before. So he really wanted that experience. And then once he got it, that would sort of satisfy him. So he would never do such a study ever again. So, it's really interesting to think, how much do we need to judge people's motivations in advance and make sure they have the right motivations? And how much do we just need to make sure that they understand what it is they're getting into and can weigh these things for themselves? And then one last thing I'll mention, happy to keep talking about the ethics, but I want to hear from Susan too, is in India right now, they're developing guidance for controlled human infection studies. And one of the key points in the initial draft guidance was to require that people do it for altruistic reasons. So they wanted to try and make sure that people were being altruistic. And I think it's a really interesting idea that raises all sorts of questions. I mean, one is, why do people have to be altruistic? Altruistic people can be exploited just as easily as people who are motivated by money. And the second thing is that, how do you guarantee that if somebody tells you they're being altruistic, they actually are? So I just thought that was a really interesting thing that I'm not sure where they're going to come out on the final guidance on that one.
That's interesting. Yeah, I don't think, first of all, the short answer to the question is, I don't think there's a set amount. And I think compensating participants in studies have primarily been based on the amount of effort that's required. So I'll use some of our studies as an example. Some of them, particularly the HIV cure studies, require a procedure called leukapheresis, which is 4 hours out of your day, and not necessarily horrendous, maybe a little bit of pain stuff. So that has a sort of a set going rate, as opposed to just coming in for visits, once every 4 months. So I think you have to put the payment in the context of what is being required. For the challenge studies, I think some of the ethical dilemma, if I can use that word, is are you paying someone just for the concept of being exposed to something, as opposed to the actual work that's involved? And and then I'll say again what I said earlier. You always have to put compensation in the context of the local environment and what can be perceived. And even though I just said that was going to be the final thing. The final thing I'm going to say is you also have to make sure that the person who is consenting has the best interest, their best interest at heart. So and I think particularly about minors and other people who may be less able to consent for themselves. I think that has to be considered.
So we have 4 minutes left, and we have one late breaking question from the audience. So I'm going to pose that question, but I also want this to be an opportunity for Seema and Susan to sort of share final thoughts or something that we haven't yet covered, but you really want to just sort of bring into the conversation to have the audience think about. So the question is from Leah Cole. Any thoughts on the inclusion of an advocate in the selection of participants, such as third-party advocates used with kidney donations? Would that impact public perception of participant protections and the informed consent process for CHI and CHM trials?
Yes.
Always more advocates.
No, but there too, you have to make sure that the advocates are well informed, impartial in their view of the process as well. And then you could throw whether they get paid in there too.
I mean, one thing that was interesting with COVID-19 was there was like One Day Sooner, which was an public advocacy like group that was trying to recruit people to participate in the trials, right? So, I mean, we did see this sort of advocacy, and it is really interesting to think about the role that they do in education, but potential issues related to some of the complications and of whether or not challenge trials were really what was needed at that given time.
Yeah, I mean, I think it's interesting, right? Advocacy, One Day Sooner, I think, did a really good job of advocating for research participants in the sense that we should recognize that people sometimes do things for very altruistic reasons, and we should celebrate them for that, just as Susan was saying earlier. And I think, historically, when we think about like the standard research participant, most of the research participants we think of are people who've been exploited or harmed in research. Those are the most famous ones. There's a statue of James Phipps, the child who Edward Jenner inoculated. But I think most people think of him as, a vulnerable participant who didn't have the opportunity to be protected as he should have been. And I think we could reconceptualize research participants and start to see their critical contributions in a new light. I think the specific issue of, how do you build better protections around research participation in a way that allows more of the research participant voice to come through and their interests to really be reflected. There are people who've advocated for the creation of unions. So Holly Fernandez Lynch wrote a paper about this several years ago, and I think this idea has been coming. I was just talking to a reporter about it this morning. This idea of, should we have unions for research participants to represent their interests? And Holly was arguing, as we've all seen with the writer's strike and the actor's strike, right? There are people who have these jobs that, they're not always employed, and they come in to do the work, and then they don't necessarily have connections naturally amongst themselves. And so there are models there, but I don't know. I think I'm not fully convinced that that's what we need. I think maybe we need more research to kind of gather the perspectives of participants, understand the burdens that they're facing, and generally really focus on treating them well and honoring them for what they do in a way, and showing them respect in ways that they would appreciate. I've been working on this a little bit with Stephanie Kraft, who has some really interesting ideas about, learning from participants themselves about what respect means to them. And I think importantly is pointed out that different people have very different conceptions of respect. So we can't assume that we know what it is that people want. We really do need to do the work to figure it out.
Yeah, and let me add to that by saying that the idea of advocacy, I think, is noble, but we also have to, in the context of PHI, we have to be respectful of people too. I mean, and I say that again from my experience with some of our HIV trials, where that was one of the early questions: should we have advocates. And I can tell you that some of the participants said, absolutely, I don't want anybody else here.
Mm.
So, I mean, so I think in the end, communication is always the key, right? We have to be good about communicating our intent and communicating the risks and I guess my other soundbite is that communication is always a two-way street, and we have to listen.
Like, I think that might be the perfect place to end. Thank you so much, Susan and Seema, for such a thought-provoking and yeah, like a really interesting conversation. Thank you for audience for asking great questions. And we will see you next month when we talk about ChatGPT as a co-author. So a little bit lighter in terms of thinking about these issues, but thank you so much, Seema.
Right. Thank you.
Thank you, Dana. This was great.
Bye.
Bye, everyone.
We're done.